Hi, Diane.
Hey, good morning.
Good morning.
I can't hear you.
My
wait a second.
audio
That's
is
my
going.
fault. Now go.
How about now?
Yeah, I had a headset in from yesterday.
Okay. So let me see what I want to, I think I'm going to switch to this.
Look at you and your experience.
Yeah, well, you know, I forget, one of my kids basically told me, Mom, we don't want to look at the dishes. You know, it's like, okay, I'll work on that. So, hey, Jer.
How you doing, kid?
I'm good. So one thing I got to test out here is, you know, I have a screen in front of me that's reasonable size, and I have my laptop right here. My camera's in my laptop. So if it's distracting to have me look at my screen and not look at my camera, I can switch the stuff around.
So, but here's the thing on it. We're going to share your screen that shows your slides, and you're only going to have a little picture of you on the side. So I guess I don't think that will make that big a difference.
See, I don't think so either.
Yeah.
So let me
try it. Let's see.
Let's try it.
Hi, Jer.
Hey, kid.
How are you?
I'm doing well. How are you doing?
Doing well. Just fighting with my orthopedic surgeon as to whether or not I get PT for my hands.
Oh, geez.
Really?
I had surgery just for like trigger fingers, and there's a disagreement on how soon afterwards you should have PT. I happen to be married to somebody who strongly advocates for early PT intervention, and my doc yesterday, through her athletic trainer, told me, we'll wait and see for about a month. So I'm like, no, I don't think so. So I'm end arounding her.
So I think one thing that will help is if I, you know,
You want me to go out and get a 12-year-old? They're very good at.
Let's see. Can't tell that we're going yet.
Caroline, you're muted.
That's unfortunate.
Now you're not.
Okay. So I'll begin. And of course, I want to begin by thanking Diane for inviting me to share with you this work this morning. I'm really tickled to death to be able to show you some of these data, which are focused on the unpublished work that we've been doing. So my lab has a favorite metal, and it's iron. And I'm gonna, okay, why doesn't that want to work?
What's up? You having a cursor issue?
It doesn't want to go.
Okay, we're ready for anybody who knows.
It actually was working just fine 5 minutes ago.
Just use the arrows up and down.
And that's what I am using.
Can I just have one more request, Caroline? With your cursor, could you right click on it and turn it into a red dot, like a laser pointer? As I get older, I have a hard time tracking a black little arrow.
Well, unfortunately, my slides don't seem to want to advance.
I would just make sure that you have the slides. You're clicked back on. There you go, the right window. If you're clicked on the actual PowerPoint, it won't advance.
Okay.
Thanks, TK. Thanks, Iqbal.
All right. Now, Iqbal says if I right click, you'll see a red dot.
You can change
Sorry?
the pointer to look like a laser pointer instead of a black arrow
I
on a
am
black
sorry,
slide.
but that does not happen with my input device.
Huh?
Sorry. So I'm going to do my best here. So like I said, my favorite metal is iron. And the reason that iron is so useful in biological systems is this little reaction here where iron can either accept or release an electron. Now that virtue is also its greatest vice because in cells that have aerobic metabolism, they're continually producing hydrogen peroxide, and iron 2 and hydrogen peroxide lead to the production of damaging oxygen radicals, for example, hydroxyl radicals here. So we have to think, how do cells use iron without creating intracellular damage? And that's one of the things I'm going to be talking about today. So iron is an essential nutrient, and it's a really important one. Iron deficiency, which is globally a very common nutritional disorder, gives us anemias, neurodevelopmental defects, and increased perinatal morbidity. The flip side of that is iron overload, and as you know, iron overload is the feature of the hereditary disease, hereditary hemochromatosis. It's also a feature of iron-loading anemias like the thalassemias. And chronic inflammatory diseases of the liver also have as a feature iron overload. So why is iron really important? It's because cells use it to make these cofactors, okay? So iron gets incorporated into organic and inorganic cofactors, and you know them as heme, this guy here, iron sulfur clusters, and we have as an example a 4-iron 4 sulfur cluster here. And the mononuclear, dinuclear iron centers that are characterized of the non-heme enzymes, okay? So there are hundreds of iron proteins in cells, and we don't even know how many there really are because we keep discovering new ones. But this capacity to use iron is very important. So I just want to remind you that in order to use these cofactors, we have to have delivery systems. The last step in heme biosynthesis happens in the mitochondria, but there are heme proteins all throughout the cell. The iron sulfur clusters are assembled both in the mitochondria and the cytosol, but iron sulfur clusters can't remain stable if they're just free in solution. They have to be trafficked by carrier proteins. And finally, these non-heme iron enzymes with their iron centers have to capture this iron from what we call the labile iron pool. Okay, so the labile iron pool is the raw material that cells use to build all of these iron cofactors. It's the kinetically exchangeable, chemically reactive pool of iron, and we're mostly going to talk about the labile iron pool in the cytosol. It has been characterized with respect to whether it's iron 2 or iron 3 by several different approaches, and the consensus is that it's largely iron 2 in the cytosol, which is consistent with the redox potential and the abundance of reductants that are in the cytosol. Now, in the words of Nigel Robinson, iron and other metals are coordinated by a polydispersed buffer system. And that would consist of both large and small molecules. Now, a systematic look at the potential small molecule ligands in the cell done a few years ago by Bob Heider's group came up with the idea that really only reduced glutathione is a reasonable ligand for iron in cells. And that's because in addition to having an appropriate affinity for iron, it's also present at an abundance that's high enough to form this complex. That is, glutathione in cells is at millimolar levels. And while cysteine and citrate do have some affinity for iron too, they're present in cells at concentrations that are really too low to support the formation of a complex. That would be the small molecule that's predicted to coordinate iron. And up until recently, there really wasn't any evidence, empiric evidence, that that's really what's happening in cells, but I'm going to talk about that today. The large molecules that would coordinate iron are the iron chaperones. And these are specialized proteins that are designed to bind iron and deliver it to targets through a protein-protein interaction, okay? And I'm going to tell you about the iron chaperones that we study. Come on now. PCBP1. Okay, so we identified PCBP1 in a genetic screen looking for proteins that would enhance the iron loading of ferritin. Now, ferritin is the universal iron storage protein in cells. It is comprised of 24 subunits of H and L chains, and it forms this hollow sphere. The iron is stored in the interior. Iron enters by traversing a pore that leads to a peroxidase site in the center where the iron 2 is converted to iron 3. Okay, and when we looked in yeast cells for human proteins that could enhance the loading of this human iron storage protein ferritin, we identified PCBP1. Okay, so it's an iron chaperone, and the criteria we used were, well, does it bind iron? Yes, binds iron 2. It also binds ferritin, and it's able to transfer the iron from the PCBP to the ferritin. And we were able to demonstrate this both in vitro and in vivo in a variety of both human cells and yeast cells. Okay, so PCBP is kind of a weird name for an iron chaperone. And that's because the PCBPs are fully characterized proteins of the H and RNP family. They are RNA binding proteins. There are 4 of them, PCBP 1, 2, 3, and 4. And structurally, they're all similar. They have 3 of these KH domains, which stands for H in RNP K homology. And this is an ancient and conserved RNA binding module. In addition to RNA, they can bind DNA, other proteins, and as we found, iron. PCBP1 and 2 are highly expressed in virtually all mammalian cells. And in at least when they're expressed in yeast, PCBP2, 3, and 4 have some activity as iron chaperones for ferritin, although PCBP1 is far and away more active. And it also appears that PCBPs have specialized iron chaperone functions. So I'm just going to remind you how iron is taken up in mammalian cells. So outside the cell, iron is largely coordinated by transferrin, okay, which associates with the transferrin receptor, and that prompts the uptake into the endosome, where the iron is both reduced and released, and then the iron 2 is transported into the cell and therefore enters the labile iron pool through a transporter like DMT1, although there are also zip transporters that can move iron 2 across this membrane. Okay, now work done by Fumio Kishi's lab in Japan suggests that PCBP2 has a specialized role in associating with these integral membrane proteins in assisting iron trafficking. The report is that PCBP2 interacts with DMT1 to facilitate uptake, that it interacts with ferroportin, which is an iron efflux pump to facilitate efflux, and that it can interact with hemoxygenase, which is a membrane-bound enzyme that liberates the iron from heme, and the PCBP2 can capture the heme from hemoxygenase. So we haven't really designed experiments to test out these ideas. I can tell you that in some of the cell types that we've been working with where we have used siRNA to knock down PCBP2, we find that DMT1 still transports well. There's not a problem with iron import. And in some cell types, at least, the iron efflux also works just fine. So the extent to which you must have PCBP2 in cells to carry out these functions is not entirely clear and may be different depending on the system you're looking at and the cell type you're looking at. But one thing that is clear to us is there's definitely interaction between PCBP2 and PCBP1, either directly or indirectly. They do both have access to the labile iron pool. Okay, so work that we have done over the years indicates that in addition to ferritin, PCBP1 is involved in delivering iron to the prolyl hydroxylases. These are mononuclear iron enzymes that regulate HIF, okay, and to an enzyme that has a dinuclear iron center, and this is called deoxyhypocene hydroxylase, which is hard to say, but is a prototypical oxo, or actually peroxo bridge diiron enzyme that needs to acquire its iron cofactor with the assistance of PCBP1. Okay, so a few years ago, we wanted to know, are there any other components to this iron distribution system? And the question that has been dogging us for a decade is, yeah, how does PCBP1 coordinate iron at the molecular level? So I'm going to shed some light on both of these questions today. So the study we did was a proteomic analysis of proteins that interact with PCBP1. We did this in collaboration with James Wolschlegel, and we pulled down a whole bunch of proteins, and one of them caught our eye, and that is BOL-A2. Okay, so you maybe never heard about BOL-A2. Before we started looking at it, it really hadn't been studied in mammalian cells, but it had been studied in yeast and plants, and we were interested in it because BOL-A2, which is the cytosolic BOL-A isoform or protemer, interacts with the monothioglutarodoxin GRIX3 and can form this iron sulfur cluster structure that keeps this complex together, okay? So this bridging 2 iron 2 sulfur cluster has a glutathione as one of the ligands, and it's bound, the glutathione's bound to GRIX3. And what has been shown by us and by others is that this is actually an iron sulfur cluster chaperone.
Caroline, will you go back one slide? We have one question.
Sure.
Which is, what determines specificity of PCBP1-2 complex to target proteins? This will be unlike the precedent set for copper chaperones. And then asked about the stoichiometry.
Okay, so I've got PCBP1 and 2 in this picture. What we know is that if you knock down PCBP2, PCBP1 is perfectly good at delivering iron to ferritin, and it is also actually delivering more iron to ferritin. So the PCBP1 and 2 are not dependent on each other for activity, but the presence of the other somehow modulates the activity. And I think it's because PCBP1 and 2 are competing for the cytosolic labile iron pool. Now, the specificity, I think, is not really so not clear to me at all, because PCBP1 and 2 are very similar. You know, the relevant structures in both are quite similar. And so what accounts for this selectivity of why you need PCBP1 to largely get this done, and why we can detect protein interactions between PCBP1 and these targets, but we don't really detect much in the way of interactions between PCBP2 and these targets, it may have to do with the affinity for iron, because all of these interactions are iron dependent. And if in the competition for iron, there is a slight advantage by PCBP1, it means that PCBP2 may not be metallated, in which case it won't have much activity, but the PCBP1 is metallated and has activity. But this is not something that is clear to me. And so if it's not clear to you, there's a good reason for that.
So if you overexpress 1 or 2 or 3 or 4, do you shift the balance and lose metallation capability?
So I have not asked the question the way you describe it. So like if I have overexpressed PCBP3, do I interfere with the capacity of PCBP1 to bind metal? That I don't know the answer. What I do know is that if you overexpress PCBP1, you can find that you form more of the complexes between PCBP1 and their targets. I think I don't have those data to show you, but and if you knock down PCBP2, you form more of the complexes between PCBP1 and its targets. But beyond that, I don't think I have an answer for you.
One more follow-up question from Paul Segala. Is the micromolar affinity of the PCBPs consistent with estimates of cytosolic labile iron concentration or labile
The
heme?
short answer, the short answer is yes, and I'm going to show you some of that data.
Great. Let's move on. Thanks so much.
We're rolling. Okay, so this is why we were interested in Bolay. Okay. And one particular, okay, and this is why we were interested in Bolay, because it's an iron sulfur cluster chaperone, and has the capacity to transfer clusters to CIAPIN1, which is an early component of the iron sulfur cluster assembly apparatus in cells. But how the iron sulfur cluster gets formed on Grix Bolay is not entirely clear. They're both in the cytosol. And so, you know, that was something that we thought, this is interesting, an interaction between our iron chaperone and this component of an iron sulfur cluster assembly. So I was not sure what the best way to share all these data with you. So I'm going right to the take-home message. This is a model of a structure that we think occurs in cells that allows PCBP1 to coordinate iron and allows this PCBP1 iron complex to coordinate with Bolay2, okay? One thing that we have learned is that to interact with Bolay2, you actually don't need full-length PCBP1. You can just do it with the third KH domain. And we've also found that this KH3 domain of PCBP1 binds both glutathione and iron, and that this forms a bridging ligand with Bolay2, and that we believe this iron-containing complex is transferred to the BOLA-GRIX3 when it's building an iron-sulfur cluster. Okay, so just to remind you, PCBP1 has 3 KH domains. This is a crystal structure of the 3rd KH domain, and it's showing you what we have identified as residues that are involved in binding a molecule of glutathione and residues that are involved in coordinating iron. And as you can see, as we've modeled it, the thiol on glutathione could also contribute a ligand to coordinate the iron in this domain. And we learned all this by doing both cell-based and in vitro assays, mutagenizing all these residues on the KH3 domain. And what you can see is, you know, with the glutathione site here and the iron binding residues here, if you mutate the 2 residues that are thought to be direct iron ligands, you don't really interfere with glutathione binding at all. But if you interfere with the glutathione binding residues, you can't bind either glutathione or iron. And so this is part of the kind of evidence that we accumulated to tell us that the glutathione is required to coordinate iron on the KH3 domain. Okay, so Paul, this is for you. We looked at the binding affinities of the KH3 domain for glutathione and found that if you looked at that, you got an affinity of around 400 micromolar unless there is iron in your buffer, and then the affinity drops down to 80 micromolar. And so this is clearly a concentration where glutathione would expect to be occupying that binding site in the cell all the time, because the glutathione level is, say, between 2 and 10 millimolar in the cytosol of cells. And similarly, when we looked at iron binding, and this is in the wild-type KH3 domain, we found a dissociation constant of 4 micromolar. And if you add glutathione into the buffer, that drops down to less than 1 micromolar, which again would suggest that this complex could form in the cell at the concentration of iron 2 that's predicted to be in the labile iron pool, which is around 1 to 2 micromolar. So we focused on this because of an observation we made in our cell-based studies. So I told you that this GRIX-BOLA complex here absolutely requires an iron-sulfur cluster to form. And if there's no complex, if there's no iron-sulfur cluster, there's no complex. So that's here. So experimentally, it looks like this. What we found is that if we got rid of PCBP1 by siRNA in our cells, the BOLA was there, but the GRIX no longer formed a complex with it, meaning that there was no iron-sulfur cluster. And in fact, you could rescue that phenotype by adding back a full-length copy of PCBP1, and there's GRIX in the complex, or you could rescue that phenotype by adding just the KH3 domain. And that meant that we could do all the studies that allowed us to define the iron site and the glutathione binding site. And so this is our model. Yeah.
Caroline, what do you think is different in yeast where you don't have PCBP and you form an iron sulfur linked GRIX3 and BOLA?
So the difference is PCBP1 is coordinating the iron glutathione complexes and reducing the reactivity of the labile iron pool. So I think what PCBP1 is doing is making this process more efficient. and allowing the cells to maintain a pool of iron in the cytosol without causing the oxidative damage that might occur if it didn't have PCBP1 coordinating the iron glutathione. Because you're absolutely correct. Yeast don't have PCBP1 and they get this done just fine. What I would point out is that even in mammalian cells, when we do this in cell culture, we find that if you flood the cells with extra iron, so if we just put a whole bunch of iron in the media, you can bypass this requirement for PCBP1. And so you could certainly form this from the bole and the Grix using just the iron glutathione. Did I cover that? Yep.
Wonderful.
John,
Okay.
do you want to ask? Sorry, Carolyn. John Phillips has a question. Do you want to ask it, John, or you want me to read it?
Oh, that's all right. I'll ask it. So is the concentration of glutathione the same in yeast and mammalian cells?
So I've seen the measurements for mammalian cells, and I haven't seen the measurements for yeast cells, but the report I see is that it is similar, that it's millimolar in the cytosol.
So that's not an explanation for it then?
I think that the iron in a yeast cytosol is also an iron glutathione complex. You know, it is interesting. Michelle Toledano did some studies a few years ago looking at the role of glutathione in yeast and came to the conclusion that yeast need glutathione to build iron sulfur clusters. And its activity as sort of a redox buffer is secondary. Okay, can I do this? So now that we have identified the iron and glutathione binding site in KH3, we could do homology modeling to try and find the corresponding sites in KH1 and KH2. And nice work done by Sarju Patel. That is what I'm going to, no, let's do that, okay. Has identified a conserved arginine residue that is required for RNA binding, as shown here, in each KH domain, and that conserved glutamate or aspartate residue that is involved in coordinating iron in each KH domain. Interestingly, the cysteine residue that coordinates the iron in KH3 is not conserved, which might explain why only KH3 will serve to facilitate iron sulfur cluster assembly on Grix bole, but KH1 won't do it. So what he has done is he's mutated these residues in each KH domain and generated versions of PCBP1 that can either bind iron but not RNA, or bind RNA but not iron. And he's expressed these in HEK cells, and what you can see is that they fold, they're stable, we can pull them down. He's looked in vitro at their single-stranded DNA binding activity, and this is a gel shift assay. And I just want to point out to you, if this is our probe, and then when we add in a wild type PCBP1, you know, we shift all the probe. But if we add in a PCBP1 that lacks the RNA binding residues, the probe does not get shifted, so it doesn't bind single-stranded oligonucleotide. And if we add in PCBP1 that has the iron sites mutated, it binds RNA just great. In fact, better than the wild type. And simultaneously, if you express these in cells and label cells with iron 55 and then pull down the PCBP1, you find iron bound to the wild type, you find iron bound to the RNA binding mutant, but you don't find any iron bound to the iron mutant. So he's made mutant forms that can separate the activities. And now we have the tools to go forward and ask which of the activities that we have attributed to PCBP1 knockdown can be directly accounted for by these either RNA or iron binding capacity. Now, I will tell you that the iron binding mutant won't bind ferritin, it won't bind bole, and I can tell you that the RNA binding mutant won't regulate the stability of transcripts that it has been reported to bind to. So we have both in vitro and cell-based data that says these mutants really are doing what we say they're doing. So we just turn to a really simple phenotype to ask the question, which of these activities of PCBP1 accounts for the loss of viability of cells when you knock it down? Okay, so this is cell-based work. We use a PCBP1 siRNA to knock down the endogenous protein. And that's the bar you see here in yellow. Those cells have greatly reduced viability. We then turn back on either an siRNA resistant version of the wild type, which is what you see here in the blue bar, not in the empty vector, that's our negative control. But if we turn back on the wild type, you can see that that completely rescues the viability of these cells. So what happens with mutants? So it turns out that it is the RNA binding activity that is required for viability. And this is in a cell culture model. We're investigating other phenotypes, and we are also planning experiments where we're going to introduce these into some of our animal models to try and look more carefully at these phenotypes. Okay. Speaking of animal models, so I've showed you some work that suggests that we have an understanding about how PCBP1 is coordinating with the labile iron pool. In fact, PCBP1 iron glutathione complexes would be the major component of the labile iron pool. And now we want to ask, well, is this actually important in an animal? Because we know that the cells and tissues in an animal are really very different than the cells that we grow in our incubators, in our petri dishes. So the first animal I'm going to tell you about is work that we have done looking at the deletion of PCBP1 in hepatocytes. Okay, so the liver is a tissue that is specialized for iron handling, because all of the dietary iron that gets absorbed goes straight to the liver. It's the major site of iron storage. It's a regulatory center because the regulatory hormone hepcidin gets made in the liver. And it's metabolically very active and has tons of iron enzymes. Okay, so we knocked out PCBP1 in hepatocytes with an albumin-Cre promoter.
Caroline, can I stop you for 1 minute? We have a couple questions.
Yep.
Dennis, do you want to ask yours? Or I can read it. Does the iron binding mutant bind different sets of RNA transcripts? If you take that off, is that a regulatory piece?
That is so interesting. So now that we have developed this tool, we can ask this question. I do not know. So one of my, and you know, the people in my group will have to just forgive me for this. One of my pet hypotheses that I'm interested in testing is asking the question, is there a strong evolutionary pressure to put RNA binding and iron chaperone activity on the same molecule? That is, are there activities in the cell where having these activities on two separate molecules doesn't get the job done? It really needs to work in cis, it can't work in trans. And so that's one of the questions that we want to ask. So I can tell you that cysteine residue that is present on KH3 was mutated in the context of studying how PCBP1 interacted with transcripts of the folate receptor. This was work done by another laboratory. And they found that that cysteine residue under folate deficiency gets homocystellated, and that affects the RNA binding activity, making it bind RNA more. And they also looked at what happens when you substitute other sort of thiol compounds like glutathione, and they also put iron into the mix. And they found that iron and glutathione could also affect the capacity to bind the folate receptor mRNA. So there may be more examples like that out there, but I haven't, investigated those. I can't tell you about them yet.
One other question from Iqbal is, have you looked for other KH3 analogs that could be masquerading as iron chaperones?
So, PCBP2 has all of these conserved residues. We don't yet know whether PCBP2 can bind glutathione with the same sort of affinity, but it's got the acidic and the cysteine residue in the KH3 domain, so it should be able to at least provide those iron ligands. But whether or not its affinity is in the same range as the PCBP1 KH3 would depend on how well it coordinates the glutathione, and we don't know that yet. H and RNPK has a KH domain. PCBP3, PCBP4, when you express them in yeast cells, they definitely turn on the iron deficiency response. So I think multiple of these domains has the capacity to bind iron, but whether they're expressed in the cell at a level that's high enough to form a complex is not clear, and whether they are interacting with other proteins or with nucleic acid in a way that sort of sterically interferes with metal binding, that I also don't know. So there's a lot of possibilities.
Great, thank you. One more. Sorry, you don't get to complete your talk. Why would an RNA binding mutant have a growth phenotype if the cell has multiple PCBPs? That's from Dennis.
I have no idea. It makes no sense to me. Because when it comes to binding RNA, you really can't distinguish between PCBP1 and PCBP2. Their RNA binding activities look virtually identical. So it must be the capacity to interact with something else, because, their RNA binding activity, they're just an adapter. They bring the RNA to some other protein complex or some other site in the cell for processing. And so it may be that the protein-protein interaction of the RNA-bound form of a PCBP determines, its specificity. But I have no idea why a cell that expresses two paralogs that are 81% identical and have identical RNA binding activities should have one of them. Well, they're actually both essential and they won't complement each other.
Okay, thank you. Sorry to stall there for a minute. Good questions.
No. It's nice to know that I'm not just having this conversation with the electrons on my monitor. All right, so mouse, liver. We knocked out PCBP1 in hepatocytes. What happened? We found that these hepatocytes become iron depleted. Okay, so there's less iron in the hepatocytes, and there is less ferritin, and the protein that triggers ferritin turnover goes up. That's NCOA4. So this is a picture of iron deficiency in a PCBP1 depleted liver. There are some, not all, but some iron enzymes that also show loss of activity. An example would be xanthine oxidase, which has a pair of two iron, two sulfur clusters. Another example is the prolyl hydroxylase that regulates HIF. Its activity goes down and you get an accumulation of HIF protein in these cells. So more of a picture of iron deficiency. What I'm not going to show you is that the iron regulatory proteins, RP1 and RP2, show the opposite picture. They act as if there's excess iron in these cells, which is crazy, but let's think about that. These mice develop fatty liver disease. So as you're probably aware, non-alcoholic fatty liver disease is a big public health problem in the United States. You can see it just by looking at a stained section of the liver, because these sort of empty little balloons were actually loaded up with fat before it went through the tissue processing. So you can easily look at a wild type liver, see healthy hepatocytes, and you can look at a PCBP1 knockout liver and see that the hepatocytes are just loading up with lipid. And we can measure that directly by measuring triglycerides. We can measure it by looking at cholesterol esters in the liver, which are also elevated. There's also hepatocyte damage happening. And we can measure that most simply by measuring ALT in the serum. Hepatocytes, when they die, release their contents into the serum, and you can measure it. And that's what this enzyme is, a measure of hepatocyte cell death.
Question.
Yep.
From May Coy about what's the age of the mice, or how long does it do? If you harvest them young, are the animals already showing fatty liver?
We've looked at several different ages. These mice that I'm showing were at 3 weeks of age, weaned onto a defined iron diet. And in this case, it's 50 ppm iron, which is sort of the basal level of maintenance. It's not a generous amount of iron by any stretch of the imagination. And after 14 to 18 days on that diet, this is what their livers look like. So these animals are roughly 5 weeks old, young. Now, if you put them on a diet that is not a defined iron diet, not a synthetic diet, but more like a natural diet, like the NIH3 diet that has basically grains and seeds, this fatty liver phenotype is delayed a bit. It takes a couple of months longer before it shows up. But you know, this is present in a young animal.
Thank you.
So, you know, these mice are iron deficient. They have steatosis. So we know that well, the livers are iron deficient, and the livers develop steatosis. And we know that there's a lot of iron enzymes involved in lipid metabolism. And so we asked, well, maybe they just need a little extra iron. So we put them on a diet that had 1,000 ppm iron. Not a ton, not something that causes toxicity in any kind of setting, but something that will certainly replete all the iron stores. And that did not help them at all. The steatosis was there, the triglyceride accumulation, and the serum ALT indicating liver damage was still elevated. So we thought, okay, well, what if we put them on a low iron diet? And so we put them on a 5 ppm diet, and they got better. The triglyceride accumulation came down, and the serum ALT came down. So that really suggested to us that without PCBP1, the iron in the liver is toxic, even if there's less of it. And so what we found, and there's lots of data that I don't have time to show you, is that unchaperoned iron in the liver is producing reactive oxygen species that cause damage to lipids. This is a stain looking at 4-hydroxynonenal, which is an end product of lipid peroxidation. And as you can see, there's quite a lot of it in these tissues from the PCBP1 knockout animals. If you directly measure oxidized triglycerides, you can see that they're elevated. And if you directly measure oxidized cholesterol esters, those are also, the oxidized forms accumulate. And so we have developed this hypothesis, we have a fair amount of good support for it, is that what PCBP1 is doing in these hepatocytes that's most important is protecting them from iron catalyzed reactive oxygen species. And so the idea being that in a wild type hepatocyte, PCBP1 coordinates the labile iron pool, reducing its reactivity, keeping it out of mischief. So this measure of chemical reactivity is down, and the accumulation of oxidatively damaged lipid is down, and you don't get steatosis. But if you take away PCBP1, you have unchaperoned iron. It's very chemically reactive, and it causes oxidative damage, and the cell reacts to the oxidative stress by increasing lipid production, and you get the steatosis. You can block this effect of PCBP1 depletion by either putting them on a low iron diet or by putting them on a diet that contains a lipophilic antioxidant, vitamin E. And what I haven't showed you are the data that support the idea that vitamin E can suppress these phenotypes as well.
2 questions. One from Rick Eisenstein asking if you've looked at mitochondrial function in these hepatocytes.
So the answer is, and this is work that Shamala Jadhav is doing, the answer is yes, we have. So some interesting things when it comes to mitochondria in these livers. Number one is, while the cytosol gets iron depleted, the mitochondria do not. The mitochondria have wild-type levels of iron, and there's no evidence that iron-sulfur cluster activity in the mitochondria is diminished. However, if you look at oxygen consumption in the respiratory chain, like using a seahorse analyzer, what you find is that there's suppressed oxygen consumption, and there are also a fair number of changes in the mitochondrial lipids. Sort of suggests that the lipid peroxidation that's happening in the cytosol is affecting mitochondrial lipids and affecting mitochondrial function. But we're working out the details on that.
So there's one other question from Dennis, which is, can you overexpress PCBP1 in a delta SOD yeast strain and suppress the reactive oxygen species problems?
Oh, I have no idea. I mean, could we? We could overexpress it. Would it have that effect? I don't know.
So you pulled it out of the screen in yeast, right? For iron loading of ferritin or something,
So what we did was we put an iron-dependent reporter, which was an AFT1 promoter hooked up to a history gene, and then we expressed heterologously ferritin, human ferritin H and L chain. Yeast have no ferritin. And then we used a library of human liver mRNA that had been engineered into a yeast expression vector. And then we screened that library for things that would move iron into ferritin and turn on the iron reporter. So that was how the PCBP1 was identified in yeast. We didn't actually try and interrogate the yeast genome to see whether there was something inherently in the yeast genome that could have this effect by overexpression. We didn't do that experiment.
Okay, thank you.
Okay, well, I don't have much time left, but I do want to share with you just a little bit of data that comes from our latest animal model. okay? And that is to look at another tissue that's very highly specialized for iron. That would be the intestinal epithelial cell. Okay, so if you think about it, it has to do a whole lot of uptake, because that's where all of the dietary iron is coming through. It has to do a whole lot of efflux, and this is all through ferroportin, right, to get the iron into the systemic circulation so the animal can use it. And I will also argue, because we have a fair amount of evidence of this, that the capacity to store iron in ferritin is also an important function of the intestinal epithelial cell. And if you lose that, you have problems. So the question that we are asking, all right, I just wanted to remind everybody that the activity of ferroportin is regulated by hepcidin that is secreted by the liver. And in models that have been looked at thus far, the efflux activity at the intestinal epithelial cell is proportional to the amount of ferroportin that sits on the surface, okay? So when the cells need more iron uptake, there is more ferroportin on the surface and there's more iron transport. Now we're gonna ask, what happens when you mess with PCBP1 and PCBP2 in this cell? Specifically, do they have any role to play in this iron flux? All right, so we knocked out PCBP1 in the intestinal epithelial cell, and this is just a DAB stain. So you can see that there's lots of PCBP1 in the crypts. It's also expressed in this epithelial cell layer, okay, on the villus. And if we go look in an animal with knockout of PCBP1 in the IEC, you can see the crypts are paler and that you don't see that brown stain on this layer, but you still see PCBP1 in the stromal cells that support this epithelial cell layer. So when you collect the epithelial cells and look by Western blot, PCBP1 is gone. So what happens? So similar to what we saw in the liver, the non-heme iron in the IECs without PCBP are greatly iron depleted. And that's true on a standard iron diet and even on an iron-enriched diet, okay? They basically are not putting iron into ferritin, and the ferritin is getting turned over. I don't show you this here, but the NCOA4 levels are really high in these cells. And so you can see by Western blot that the ferritin is pretty much gone in the PCBP1 knockout IECs. But that doesn't mean that iron is not getting into this animal, because if you look at iron in the serum, it's either normal or elevated in our knockout animals, okay? And if you look at iron in the liver, it's either normal or elevated in the IEC knockout animals, which suggests that dietary iron is being readily absorbed and made available, but it's not being retained in the IEC. Okay. So we looked to see what ferroportin was doing in these animals. And, you know, here's a villus that's been stained with an anti-ferroportin antibody. And as you can see, there's a nice strong signal. It has a basolateral distribution. And we've seen this with a couple of different antibodies. However, if we look at PCBP1, the signal is exceedingly low. And we can quantify that. We have also looked at ferroportin by Western blot, and it is again lower in the PCBP1 IEC knockout animals, although not quite as low as what you see by IHC. So how is it that these animals are transporting lots of iron to the systemic circulation, but they have very low levels of ferroportin? Well, we decided to look more specifically at the kinetics of iron absorption using an iron 57 tracer. This is a stable isotope of iron. it's naturally occurring in very low levels, and we can give purified iron 57 to these animals and measure its trafficking. So if you orally gavage this, you find that the amount of this tracer that gets retained in the IEC, and this is after like an hour, is really much less, okay? But it's not because of any defect in uptake, because the amount of the tracer in the plasma and in the liver is up, okay? So we have the capacity to measure iron transport across the IEC now. So what is ferroportin doing? So we received from Tom Gantz and Ellen Namath a really useful little peptide they called the mini hepcidin. This peptide will bind to ferroportin and block its activity. And so we could use that to give ourselves an acute blockage of ferroportin activity to ask whether the iron transport across the intestinal epithelial cell is ferroportin dependent in these animals. And so we looked in the IECs after an oral dose of iron, and we found that in animals where ferroportin activity had been blocked, there's accumulation of the iron 57, and in animals that were the control animals where it wasn't blocked, you again see that there's very low levels of iron 57 in the IEC of the knockout animals, but pretty much is retained in the wild type animals. And then if you look in the plasma, the mini hepcidin completely blocks the transfer to the plasma in both the wild type and the knockout animals, which means that even in the absence of PCBP1 and in the relative downregulation of ferroportin, this enhanced iron flux is all going through ferroportin. And we've sort of uncoupled the regulation of ferroportin via hepcidin from iron trafficking. And so that would say that, you know, PCBP1 is needed to regulate the flux of iron through the IEC and the efflux through ferroportin. So just going to summarize here because I'm all out of time. So what have I told you? That the labile iron pool is an iron glutathione complex that is bound by PCBP1. And that PCBP1 makes iron available to non-heme enzymes, that it links the cytosolic iron pool to the iron cluster assembly system via an interaction with BOLA2. In erythrocytes, and I didn't get to show you this data today, the flux of iron through ferritin, which is mediated by PCBP1, is needed for efficient heme synthesis and erythrocyte development. And in hepatocytes, we need PCBP1 to prevent iron from causing oxidative damage within that cell type. And in the intestinal epithelial cell, you need PCBP1 to control the efflux of iron through ferroportin, okay? So I just want to thank the most awesome sequestered members of my group, plus Al, who's not in this picture. Sarju has done all the work on the BOLA and KH3 domain system. An excellent team of Ethan, Olga, and Shamala are working on the liver-specific knockout. Yubo Wang has been looking at the intestinal epithelial cell knockout, and my very talented technician who does all of our genotype and keeps all the trains running on time is Minu Shukri Elise. And are there any more questions?
Great talk, thank you. I'm gonna let anybody who wants to talk ask other questions. There's just unmute yourself and chime in. That was great, really fun.
Spectacular talk, Carolyn.
Thanks.
Hey, Caroline,
I have a question.
Ella.
This is Tom Gann. Oh, go ahead, Ella.
Okay, so my question is, Do you know what may be regulating ferroportin? What is causing it to go down on the protein level? And what's the effect on mRNA level in your intestinal knockout?
So the
Just ask the
mRNA
question.
okay. So the mRNA really doesn't change much. So I can't see my yeast colleagues right now, but I suspect that they are all nodding their heads, because when we studied iron transport in yeast, one of the things that would trigger downregulation of the transporter is a whole bunch of flux through the transporter. I think that this is what's happening in the intestinal epithelial cell, that if the ferroportin is doing lots of transport, that the cell is going to try and downregulate the ferroportin, and that it's probably happening through one of the, you know, well-described mechanisms that would be, you know, tagging it with the ubiquitin and pulling it off the membrane and sending it to the lysosome. We haven't been able to measure that. That's one of the things that we were almost ready to do when the coronavirus came. And so when we get opened up again, you know, Yubo is hoping to be able to get his primary cell culture of intestinal epithelial cells going, and we'll address that question directly.
Other questions?
Caroline, I have a question again. So with the RNA binding mutant, does it bind iron acceptors as well?
When you say iron acceptors, what are you thinking?
Well, you know, the various proteins that PCBP would bind to.
So, so the RNA binding mutant interacts quite well with ferritin and bole. One of the things that we want to know is there a target protein in the cell? For example, the replication apparatus, which has iron sulfur clusters and single-stranded nucleic acids or helicases that are involved in DNA repair, where there's iron sulfur clusters and there's single-stranded nucleic acid. Do you need to have both iron and RNA binding at that point, at that site? And that's a question we would like to understand.
Yeah.
Any other questions?
Diane, there's a bunch of chats, but are those people who've already,
Yep, I think I've addressed all the chats.
okay. Hey, I didn't get a chance to give a really good shout out to my colleagues at the Metabolomics Core at CIHD, but they have done tons of work for us, including looking at specific lipids in the cell that are oxidized and wild type, and also looking at specific lipids that are in mitochondria, which we will be commenting on in a future publication, because it seems like that has been very useful in trying to understand mitochondrial metabolism in these liver-specific knockout animals. So, woohoo, to the Metabolomics Core.
Thank you.
Good response. Move on. Okay, I'm going to close the meeting now so other people can get on to other daily activities. I want to, again, thank you so much, Caroline. That was a great talk, really fun, very thought-provoking. And we have another presentation next week by Jonathan Barash, and we'll hope to see you all then. Bye.
Thanks, Caroline.
Bye.
Thank you.
That was great. Thanks.
Thanks for having me.
Yeah, also perfect pitch for the CIHD. We're going in for our renewal.
And John's going to get a letter from me, I promise.
Thank you. Thank you. I really like the regulation of ferroportin. That's very interesting.
It is. but I think maybe we shouldn't be surprised.
No, and I agree with you. The more of the substrate you transport through these transporters, they get stuck in a conformation that probably makes them more susceptible to downregulation. So that's super reasonable. Don't you think, Jer?
Yes, I do.
Didn't you guys describe that with FET3? Yeah.
Yes. Can you rescue your cultured cells that in which you downregulate PCB1? You have the, now, by the way, I might have missed it because in between this is happening, one of my granddaughters graduated from public school.
Yay.
Yay. So the phenotypes that you see in cultured cells when you knock down PCBP1, they are really magnified if the cells are just the tiniest little bit iron deficient.
Which is culture media.
Which, you know, just, you know, and you don't have to make them hugely iron deficient, you know, not iron deficient like it's going to knock out like